Hello everybody, welcome back to our medicinal chemistry class. I guess today is going to be my portion of journal club and I'm going to be focusing on antivirals, but more specifically everybody's current favorite topic, the coronavirus. The journal that I got, it's from the Journal of Virology. It's entitled Molecular Mechanism for Antibody-Dependent Enhancement, or ADE, of Coronavirus Entry, and it's by all of those authors there at the bottom, which you can read for yourself. So to begin, we're going to go over a little bit of background. The first couple slides, I'm sure you guys all know already, but I'm going to give it to you anyways. The coronavirus, they're large, positively stranded, enveloped RNA viruses. As we know, they're highly pathogenic and pretty lethal. The disease causes respiratory illnesses similar to the flu, but more severe usually. So the symptoms include cough, fever, difficulty breathing, and then in very severe cases, we've seen a lot of death happen. The coronavirus, it spreads primarily through contact with infected person when they cough or they sneeze, but you can also get it if there's the virus on some kind of object and then you touch it and you touch your eyes, nose, mouth, typical ways that a flu-like virus spreads. There's two types of coronavirus. There's the SARS and the MERS. So SARS stands for Severe Acute Respiratory Syndrome Coronavirus, and then MERS stands for Middle East Respiratory Syndrome Coronavirus. This article mostly focuses on the MERS. It talks about SARS a little bit, but for the most part, we're going to spend time on MERS. As we all know, the world is currently in a bit of a crisis due to this disease. We can see from this graph here just how severe it is. This graph is from April 1st. The number of COVID cases all around the world, it's, as we can see, rising really, really quickly. This is showing the number of days, like how many cases per day. So in the US, we're seeing 20,000 per day, which is only rising, and that's a really scary thing, but luckily we are starting to see some downshifts as well in China and South Korea, where the time since the first exposure has been longer than in the US, but at least we're starting to see a decrease in some countries. We're all doing this thing called flattening the curve. Due to this, national lockdowns have begun, and this means just basically this flattening the curve means taking extra sanitary precautions. It means social distancing. It means limiting the number of people in a gathering. Last I heard it was 10. It might be smaller now. It's basically reducing the number of people exposed to the virus and therefore reducing the number of people who fall ill, which flattens the curve and reduces the number of cases that we have overall. And so far it's proven to be a pretty effective measure in fighting the virus, but we still need a cure for it, some kind of well, not a cure, but a vaccine or a treatment, because not much is really known on the coronavirus. It's been around for years and years, but it's never reached this level of severity, so it's not been studied a whole lot. That means there's not a whole lot of research done and not a whole lot of understanding on the disease itself. So specifically, this article is looking at the mechanism of its entry into cells and how that entry is actually enhanced by antibodies. And therefore, this area of research is really important to understand how we can try to stop that. So now I'm going to talk about the structure of the coronavirus. And the rest of this is stuff you guys may not know. It's not been in the news as much. So the structure we can see in that picture there, the coiled pink on the inside there, that's the RNA, and it's covered by that red, which is the envelope, because this is an enveloped virus. The blue is the hemagglutinin esterase dimer, but what I want to focus on and what this article focuses on is the yellow portion. So the yellow is the envelope anchored spike that guides entry into the host cells. The spike gets a homotrimer with 3 receptor binding S1 units, which is why you could see how there's like 3 protrusions out of each spike. But it also has a trimeric membrane fusion S2 stalk, which is where it kind of goes into the membrane itself. So the state that it's in here on the mature virins is called the prefusion, but there's also a post-fusion state, and that's when the spike, it hits the membranes of the host cell, and so then the membranes of the host cell and the virus fuse together, hence post-fusion. That fusion, though, it only occurs after receptor binding and after conformational changes, from two cleavages of that spike. And then the spike, the S1 and the S2 portions have to disassociate from each other as well. So first the S1 and the S2 boundary is cleaved, and then the protease or the proteolysis occurs either during maturation by pro-protein convertases or after viral release by extracellular proteases, or after viral attachment by cell surface proteases, or after viral endocytosis by lysosomal proteases. So there's a lot of times when it can go through that maturation, but if you're going to get infection, that's going to happen. So it's important to notice the difference between the RBD, which is the receptor binding domain, which is the part that interacts with the protein on the cell that it infects, the host cells, and the SE, which is the ectodomain of the spike, so it's the part that sticks out, and it's the portion that inserts itself into the membranes to cause the infection. So one of the techniques that the researchers used was an ELISA, and I mean, we've learned about ELISAs in biochem. You have your capture antibody, then you block the plate so that the antigen won't stick to it and it'll only stick to your antibody. And then you wash it, you attach a second antibody, and it has a enzyme on it that will change your substrate into your product, and then based on that reaction you can measure whatever you're trying to measure for the binding affinities. And that's pretty much what they did in this study as well. They were looking for the binding affinity between the monoclonal antibody that they used and the MERS-CoV, either the ectodomain or the receptor binding domain, the RBD. We'll see this applied later. It'll probably make more sense when we get to the figure. But they did two types of ELISAs. And the first one was more normal. The ELISA plates were coated with the antibody like we expect. They were blocked, and then you had the antigen of either the MERS-CoV with the ectodomain or with the RBD, and then it was washed with PBS like normal. The plates then, they were incubated with anti-His6 antibody because the recombinant proteins, they have a 6 His tag on them, and that's identified if it's bound to the anti-6 His antibody. And then again, they were washed with PBS and incubated, this time with horseradish peroxidase, which is, it's a conjugated goat antibody. And they did that because it attaches to the anti-6 antibody, and that's what gives your color change from your substrate changing to your product. So part A was pretty normal, but part B is where it was different because they reversed the roles. So now in part B, which I'll mention later, the antigen was actually what coated the plate. It was the one in excess. And then the antibody is placed over top of it, and then it's followed by the same detection scheme with the anti-6 His6 antibody and the horseradish peroxidase. So it's a bit of a different technique, but it makes sense with their figures. Another technique that they used was flow cytometry. I think we pretty much know about this, but just as a little catch up, I guess. It's used to measure physical and chemical characteristics of cells. So what happens in this is a sample that contains the cells or the particles is suspended in a fluid and then injected into a flow cytometer. And the sample, it's focused so that only one cell goes through the flow cell at a time. And each of the cells is hit by a laser beam at different, you can change the excitation levels of that so that the laser beam, when it hits the cells, based on the size, shape, structure, component of the cell, the wavelengths are bounced off differently. And so the cells, they're often labeled with fluorescent markers so that light is absorbed and then emitted in different bands of wavelengths, which is why we have all these mirrors here, because some of the light is going to go straight into the capture, like the detection, but some of it is going to be bounced off in different angles, so not in just a straight line all the way through the cell. It's going to be bent, and so that's going to be picked up by different sensors coming off of those mirrors. Okay, so the method that I really want to talk about is the coronavirus spike-mediated pseudovirus entry assay. It's one that we don't know about. So this pseudovirus packaging, they use HEK293 T cells, and these cells, they were co-transfected with a plasmid that carried an ENV-defected luciferase-expressing HIV type 1 genome, and then a plasmid encoding either the MERS or the SARS-CoV spike. After that, the viruses were harvested and they were purified, and they did that by using a sucrose gradient ultracentrifugation. And then the cells were allowed to incubate for a little while. At one point they changed the medium and let them incubate a little bit longer. And then the cells, they were lysed open so that we could get to the enzymes that we're interested in, which is luciferase. So to the aliquots of the cell lysates, the researchers, they added luciferase substrate and then measured the relative light units, and that's really how you tell how much has entered your cell. So really, to put this in simpler terms, what they were doing, the researchers modified the virus to express luciferase, which is an oxidative enzyme that produces bioluminescence. So once the cell lysates were treated with a luciferase substrate, the enzyme oxidizes the substrate and then produces light as a result, which could be detected to determine how much the virus was able to enter the cells, which I didn't know this, it was a cool fact, but it's actually the same enzyme that causes the light up in fireflies. Okay, so moving on, just a little bit more background. Antibody-dependent enhancement, or ADE, it's when viruses access a host cell and that entry is bettered with the addition of antibodies, which is unfortunate because, as we all know, our body makes antibodies. So ADE, it's seen in other viruses as well, like the West Nile virus and like Zika virus. But primary infection by one serotype makes neutralizing antibodies to target that serotype. But then when you get a secondary infection by a different serotype, this does not allow those pre-existing antibodies to neutralize the virus in the same way that it did with the first infection, but instead the antibodies that you make when you're infected the second time, those bind to the secondary virus, and then they bind to the IgG-Fc receptors on the immune cells, and they actually allow the infection of those cells. So it's similar to HIV and Ebola. So therefore, the second antibodies that you make, called subneutralizing antibodies, they're what causes the ADE. And as we know, antibodies are super important for immunity, so ADE is a really important area for research for a lot of things, but especially vaccine design and antibody-based drug therapy. Because it's really dangerous if we have our body making antibodies against a disease, but then if we get just a different strain, all of a sudden our body is helping that disease enter instead of fight it off. Okay. And then for this slide, Sera-induced by SARS-CoV, it actually enhances viral entry to the Fc receptor expressing cells. And then SARS-CoV, it doesn't go through the same receptor-dependent pathways to enter the cells, but instead enters through viral receptor-dependent pathways that are also used by the neutralizing monoclonal antibodies. And the monoclonal antibodies used in this study, they're going to be called MERS monoclonal antibodies, and those target the RBDs of the MERS-CoV. So the monoclonal antibodies, strong binding affinity for MERS-CoV RBD, neutralizes virus entry by outcompeting DPP4, which is the natural viral receptor of the MERS-CoV, which we'll talk about here with figure 1. So the whole research question for this article was to understand that mechanism of ADE in coronavirus, in which fully neutralizing antibodies mimic the function of a viral receptor in mediating that viral entry into Fc receptor expressing cells like your immune cells. Okay, so for figure 1, this is showing the binding area, the binding sites, and the amino acids in the MERS-CoV spikes. So basically, there's two things you need to know first to understand this. SARS-CoV, it recognizes an enzyme called ACE2 as its viral receptor, but MERS-CoV, it recognizes dipeptidyl peptidase 4, which is DPP4, like I just mentioned, I think 2 slides back. And both S1 receptors, they contain a receptor binding domain to mediate the receptor recognition. So in 1A, this is just showing the tertiary structure of the MERS-CoV RBD complexed with the DPP4. So this is your natural state. And DPP4, it binds to the receptor binding motif of the RBD. In B, though, knowing the general structure, the RBD, it can have conformation of standing up or lying down. And binding to a viral receptor actually stabilizes the up state. In 1B, it shows one of the monomeric subunits of the COVID spikes in the standing up conformation, and it's complexed with DPP4, as we can see. And then the other 2 monomeric subunits are lying down and not interacting at all. And then in 1C, this is showing the tertiary structure of the MERS-CoV, like in 1A, but instead of binding to DPP4, it has those green sticks on it with the amino acids labeled. And the green sticks for the amino acids, they represent the monoclonal antibody binding residues, so where it will bind to the virus. And you'll notice that this is a lot of charged amino acids, and these locations, they were found through mutagenesis studies. So figure 1 is actually pretty simple to understand. It's just kind of for some knowledge about structure. So knowing the binding regions and the structures of DPP4 and monoclonal antibody, the researchers then sought to characterize the interactions between the antibody and the MERS-CoV spike. And this is because the MERS monoclonal antibody, it's used because it's a MERS-CoV RBD specific monoclonal antibody. So characterization was done using biochemical methods like ELISA, which this relates back to the ELISA slide that I was talking earlier with parts A and B and how it was a little bit different. So in A, the ELISA, it was done more the normal way, like I said. It was done for the MERS antibody and then the MERS-CoV RBD binding, as well as the MERS antibody and the MERS-CoV spike ectodomain binding. And the ectodomain, like I said, is just the part that sticks out of the virus and causes the interaction with the other cell and then the infection. So the monoclonal antibody, it was used to coat the ELISA plate in A, and then different concentrations of the RBD and of the ectodomain were added for the detection of their binding to that antibody. As we can see, both of them bound to the antibody, but the ectodomain bound to the antibody more tightly, which is indicated by more ELISA binding on our y-axis. The researchers claimed that this was due to the multivalent effects associated with the trimeric state of the ectodomain, and then they also tested it with PBS just to have a negative control and show that nothing binds with the control, but only to MERS. But then I was confused why they used PBS. It just didn't really seem right to me, so I talked to Dr. Miller about it, and we agreed that this is probably a critique of this paper. We agreed that a similar isotype antibody would have probably been better, and they actually do show that in another figure. but PBS works fine to show that negative control as well. Just for information's sake, though, PBS might not have been the best decision, but it's okay. Part B then, this is where the ELISA is a bit different, because now instead of having the monoclonal antibody in excess coating the plate, we have the ectodomain instead, which is a bit strange. We never learned about that in biochem. I had to ask Dr. Miller about that as well. But for this, they used the ectodomain and then they bound either the monoclonal antibody or a Fab of the monoclonal antibody to it. And again, they did that using different concentrations of the Fab and the monoclonal antibody to see how their binding was to the ectodomain. And clearly the antibody bound more tightly to the ectodomain than the Fab did, again, due to the y-axis. And then this was also thought to be due to the multivalent effects associated with the dimeric state of the monoclonal antibody. Okay, so then in 2C, instead of using ELISA, they used flow cytometry. And here they're detecting the natural binding between the ectodomain and the DPP4 receptor, and between the ectodomain or the ectodomain and the anti-MERS monoclonal antibody, or the ectodomain and the anti-SARS monoclonal antibody, all three of those with CD32A. And CD32A, it's a FC receptor. So DPP4 and CD32A, both receptors, they're expressed on the surface of the human HEK293 T cells. I don't remember if I said it on the previous slide or not, but those cells, they're human kidney cells. But recombinant SE, which is the ectodomain, it was added for detection of potential binding to one of the two receptors, either DPP4 or CD32A, in the absence or in the presence of the monoclonal antibody. And then looking at the figure, the results showed that without the monoclonal antibody, the ectodomain bound only to DPP4, but then with the monoclonal antibody, the ectodomain also bound to CD32A. This was detected through fluorescence-labeled anti-His6 antibody added to the target of the C-terminal His6 tag on the ectodomain. Cells were analyzed using FACS, which is fluorescence-activated cell sorting, just flow cytometry. And then as a negative control, the SARS-CoV RBD-specific monoclonal antibody was used, and it didn't mediate the binding of the ectodomain to the CD32A, as we can see. Alright, so then 2D, which is easier to understand, is just the Western blot, and it's showing the expression levels of cell membrane associated DPP4 and CD32A. And D and C actually relate to each other, and D was used to normalize C. So this was done by, again, targeting the C-terminal tag. And the results of the Western blot, they show that there's direct binding of the ectodomain to DPP4, and that it's stronger than the indirect binding of the ectodomain to the CD32A. As a control here, they used actin, and the actin antibody, it's used to measure the expression level of cellular actin, and they used actin because it's a loading control. So it should appear the same no matter what cell type is used, like the thickness of the band on the Western blot. And so if it does appear the same, then you know the amounts that you loaded are correct. So again, just your loading control. And then figure 2 overall ABCD. It's showing that the monoclonal antibody directly binds to the RBD region of the MERS-CoV ectodomain, and it also mediates the indirect binding interactions between the ectodomain and the FC receptors on cells. Okay, so moving on from figure 2, the researchers, they then wanted to test if the monoclonal antibody mediates the MERS-CoV entry into the FC receptor expressing cells, not just the binding of them and the interaction of them, but the actual entry. And to do this, they use that MERS-CoV pseudovirus entry assay that I talked about. So like I said in the entry assay, the pseudovirus, they transfected it with luciferase, and then it allowed the researchers to focus on that viral entry, because when you add the luciferase substrate, if your entry was successful when the cells were lysed open, the luciferase can act on the substrate and cause the fluorescence, which is what they were measuring, and that's how you get your y-axis. So the efficiency then, it was measured using the luciferase, the efficiency of entry, I should say. And the researchers, again, they used these HEK 293 T cells, as we see. And these cells, they express the Fc receptors on that x-axis. So that's the CD16A, the CD32A, and the CD64A. And then they also use macrophages, and that was because the macrophages have mixtures of Fc receptors that are endogenously expressed. And the MOC, that's a control, so those are the cells not expressing viral receptors or Fc receptors on them. And then as we can see in 3A, no antibody prevented the MERS-CoV pseudovirus from entering the Fc receptor expressing cells as expected. But with the antibody, the MERS-CoV pseudovirus entered CD32A expressing cells and the macrophages successfully. In contrast then, with no MERS antibody, the MERS-CoV pseudoviruses, they were able to enter the DPP4 expressing cells, but the antibody presence caused a highly reduced entry into those cells as well. So according to the research and the control experiments, the anti-SARS monoclonal antibody, it didn't mediate the MERS-CoV pseudovirus entry into the Fc receptor expressing HEK 293 T cells or in the macrophages, and it didn't block the MERS-CoV pseudovirus entry into the DPP4 receptor expressing cells either. In B, then, 3B, they're kind of doing the same assay, but this time the experiment, it was run the same as in A, but only the Fc and the Fab portions of the antibody were used. So here the researchers, they're demonstrating that neither the Fc nor the Fab portion of the antibody could mediate that entry into the Fc receptor expressing cells or the macrophages, suggesting that both the Fc and the Fab portions of the anti-MERS monoclonal antibody are required for the viral entry success. Okay, so moving on to 3C, the researchers here, they were then testing the level of DPP4 in different cell lines. So first, they looked at the DPP4 expressing HEK 293 T cells, and these cells, they were induced to exogenously express high levels of DPP4 to detect background expression levels, and they were able to extract the RNA and use reverse transcription quantitative PCR. And then the expression level of the DPP4 was defined as the ratio between the RNA of DPP4 and the RNA of GAPDH. So from figure 3, we can see that the HEK 293 T cells, they express very low levels of DPP4 compared to the cell line MRC5, which are human lung cells, and they express just a little bit more DPP4 than the HeLa cells, which are human cervical cells, which don't express any DPP4 at all, which is fine because the HeLa cells are actually used as the control here. And that's really all the 3C is saying. So moving on to 3D, at this point, the researchers claim pretty much directly from the text that due to the amount of comprehensive control experiments that they had, the very low endogenous expression of DPP4 in the HEK 293 T cells shouldn't affect their conclusions, and so the results from figure 3C are okay to have. But to prove this, they showed figure 3D in that HeLa cells that do not express DPP4 still have antibody-mediated MERS-CoV pseudovirus entry, meaning that the MERS antibody mediates MERS-CoV entry into the Fc receptor expressing cells, but blocks entry into the DPP4 expressing cells. And then 3E further proves this point, basically. So here it's the exact same experiment as in 3A, but now they're using SARS coronavirus instead of MERS coronavirus. And because they're doing that, now they have to use a SARS coronavirus RBD-specific monoclonal antibody, and that one is called 33G4. And 33G4 binds to the ACE2 binding region of the SARS-CoV, which is the difference that you see there on the x-axis between 3A and 3E. So 3E was testing for the ability to mediate the SARS-CoV entry now. And as we can see, 33G4, it mediated the SARS-CoV pseudovirus entry into CD32A expressing cells, but blocked the SARS-CoV pseudovirus entry into ACE2 expressing cells. It meant that both the MERS-CoV RBD-specific monoclonal antibody and the SARS one mediate the entry of respective coronavirus into the FC receptor expressing human cells, but blocks the entry of the respective coronavirus into viral receptor expressing cells. Okay, so next the researchers wanted to discover the molecular mechanism for antibody-dependent enhancement, ADE, of coronavirus entry. So to do this, the researchers, they wanted to determine if the antibody caused the conformational changes of the spike that allow that to happen. And like we talked about earlier, DPP4 binds to the spike and stabilizes the RBD in the up position, but it also results in weakening of the spike that allows for the S2 to get cleaved. This was actually shown in a previous study by Millet et al. In 2014. So the researchers of this study, they basically just replicated that, and they used the MERS-CoV pseudovirus and incubated them with DPP4, and then exposed them to trypsin cleavage, which is shown in 4A. They used an anti-C9 tag monoclonal antibody to detect the C-terminal C9 tags on the cleaved spike, which, as we know, is important for actually getting bands on a Western blot. You have to have something to detect, so that's what they were detecting. The figure, it shows that the MERS-CoV spike molecules, they're cleaved at the S1, S2 site by pro-protein convertases in all cases, in the MOC and the PBS and the DPP4 anti-MERS monoclonal antibody and in the anti-SARS monoclonal antibody. So that first cleavage happens in all cases that they tested. But the spikes, they underwent the second cleavage at the S2 site, which is labeled S2 prime in figure 4A, if DPP4 was present. So from this, they suggested that the S1, S2 site was accessible to proteases in the free form of the spike, but DPP4 binding triggers a conformational change of the spike that exposes the S2 site for cleavage. And it's pretty much only the DPP4 that allows that, but as we see, they also showed that the MERS antibody binding also allowed for that to be cleaved. So it's only DPP4 and anti-MERS antibody that allow for that conformational change of the spike to get cleaved and allow for that maturation to occur. For their controls, the SARS-CoV RBD specific monoclonal antibody served as the negative control, and it obviously didn't trigger the spike to be cleaved at the S2 position since there's no band at the 72 region. This just means that the monoclonal antibody for MERS is also what triggered the conformational change and nothing outside of what they expected. So then moving on to 4B, this figure is interesting. I don't think Dr. Miller liked it. I know I don't like it. So this figure, it's trying to show, I should say, the negative stain electron microscopy, EM, for binding between the monoclonal antibody and the MERS-CoV ectodomain. So the researchers, they used FAB in the EM because they had detected the binding of the FAB and the ectodomain in this figure. The EM showed that the FAB binds to the tip of the SE trimer where the RBD is located, as shown by the arrows. I don't know about you, but I can't see this, so I didn't know I don't know how great of a figure that this is, but what they said in the article, they did note that the limited resolution did not allow them to see the conformation of the Fab-bound RBD, but they knew from previous studies that the receptor binding site on the RBD is accessible only when the RBD is in the standing up position, and that it's most likely the case here. So I kind of feel like based on the previous studies, they can safely say that the Fab binds to the tip of the ectodomain trimer. The power of this figure, according to Dr. Miller, is that you can look at the different orientations of this binding. I don't really understand how they got that from this figure, but apparently if you take individual particles, you can make a model with high resolution that shows those interactions. So overall, figure 4 then is just trying to show that DPP4 and the MERS monoclonal antibodies stabilize the RBD in the standing up position and triggers that conformational change of the spike. We're going to skip then to figure 7 because I was saying that Dr. Miller said that from the EM you can get a model. This is the model. It's not actually from this study, it's actually from a previous study. But it shows how antibodies interact with the SARS and with the MERS, and that the antibody allows that conformational change. It also supports their data. So in 7a, it's showing a SARS-CoV RBD specific monoclonal antibody, which is that S230, and it's showing that it binds to the ACE2 binding region and stabilizes the RBD in the standing up position and triggers the conformational changes of the spike. And then 7b, it's showing the MERS, and that its monoclonal antibody, in this case is the LCA60, it's binding to the side of the RBD away from the DPP4 binding region, stabilizing the RBD in the lying down position, and it doesn't trigger any conformational change. Okay, so figure 5, they then wanted to show the impact of different proteases on the MERS-CoV pseudovirus entry. So proprotein convertase inhibitor, PCI is what I'll call it from now on. It was used to show how that impacted maturation of the MERS-CoV spike and then its impact on resulting monoclonal antibody-dependent viral entry as well. So 5a, it uses the MERS-CoV pseudovirus entry assay, like in figure 3. That's how we get our y-axis again. But when we look at this next result, I was confused originally, so I'm going to go through this really quick. We're not actually comparing the clearly visible bars to each other, so like not the dark bar and the dark bar and then like medium gray and medium gray together. We're comparing the bars in the presence and in the absence of the antibody, which is why I have those red boxes in there to kind of help the understanding, because I was confused on it for a really long time, so hopefully that'll help you guys. But for 5a, this first part that I have boxed, it's to show that without the monoclonal antibody, the MERS-CoV pseudovirus entered the DPP4 expressing cells more efficiently for the viruses with PPCI than those without PPCI. And then, there we go. It also showed then for cells that showed the antibody that the MERS-CoV pseudovirus entered CD32A expressing cells packaged in the presence of PPCI more efficiently than those without it. Moving on to B then, we see that from the Western blot that when the pseudoviruses were produced from the HEK293 T cells, the cleavage of the spike by protein convertases was really inhibited by the presence of PPCI. So then from 5A and B, we know protein convertases play a role in both the DPP4 dependent and the monoclonal antibody dependent entries. Okay, so 5C, it's not as intimidating as it looks actually. In 5C, they're looking at cell surface protease TMPRSS2. It's a transmembrane serine protease 2. And it's tested for its role in monoclonal antibody-dependent viral entry. So we see that without the MERS monoclonal antibody, the TMPRSS2 enhanced the MERS-CoV pseudovirus entry into the DPP4 expressing cells. And then here, with the monoclonal antibody, TMPRSS2, also enhanced the entry into CD32A expressing cells. So the results of 5C indicate that TMPRSS2 activates the MERS monoclonal and antibody-dependent MERS COVID entry. Okay, in 5D, we're looking at lysosomal protease inhibitors. So we're looking at lysosomal acidification inhibitor, which is BAFA1, and then cysteine protease inhibitor, ED4D. And these were tested for their role in the MERS antibody 1 dependent viral entry. And to kind of clear up this figure, I put what we're looking at in the red boxes. So both lysosomal protease inhibitors, they blocked the DPP4 dependent viral entry pathway, as we can see comparing it to that first dark black bar. And then looking at the figure as a whole, we see that the lysosomal protease inhibitors, they also block the MERS antibody 1 dependent viral entry pathway. So the results suggest that the lysosomal proteases play important roles in the MERS monoclonal antibody dependent MERS COVID entry. And figure 5 as a whole, it shows that the DPP4 dependent and then the MERS monoclonal antibody dependent MERS COVID entries can both be activated by proprotein convertases, cell surface proteases, and lysosomal proteases, meaning the same pathway is shared by DPP4 dependent and MERS monoclonal antibody dependent MERS COVID entries. Okay, figure 6, our last figure. The researchers now, they wanted to find the antibody dosages for antibody-dependent enhancement of the coronavirus entry, just to better understand it. And the MERS monoclonal antibody dosages, they were found using that entry assay again, but in the presence of different concentrations of the monoclonal antibody. So 3 types of human HEK293 T cells were used. They used the cells exogenously expressing DPP4 only, C32A only, or both the DPP4 and the CD32A. So in 6A, for the cells expressing only DPP4, as the antibody increased, the viral entry decreased, meaning that the MERS monoclonal antibody blocks the DPP4 dependent viral entry pathway, and it does that because it outcompetes DPP4. However, as the MERS monoclonal antibody increased, the viral entry into the cells that express CD32A, at first it increased and then it decreased. So the researchers' hypothesis was that this occurred because at low concentrations, there's more monoclonal antibody molecules to enhance the indirect interactions between the MERS COVID spike and the FC receptor, but at high concentrations, the monoclonal antibody molecules saturate the cell surface FC receptor molecules and then bind to the MERS COVID spike and block the indirect interactions between the MERS COVID spike and the FC receptor. And then in 6B, for cells expressing both the DPP4 and the CD32A now, as the MERS monoclonal antibody 1 increased, the viral entry first dropped, then increased, and then dropped again, as we can see. So the researchers explained that when both DPP4 and CD32A are present on the surface of the cells, the MERS monoclonal antibody, it inhibits the viral entry by blocking the DPP4 dependent entry pathway at low concentrations, but that the MERS monoclonal antibody, it also promotes viral entry by enhancing the CD32A dependent entry pathway at intermediate concentrations, which is why we see the up, and then we see the down because the MERS monoclonal antibody inhibits viral entry, again, by blocking both the DPP4 and the CD32A dependent entry pathways just at high concentrations. For figure 6C and D, this time they're using MRC5 cells instead of HEK293 T cells, and they get pretty much the exact same results. And the reason that they changed the cell line and show C and D, it's to confirm their explanation for the results that they found in 6B. The MRC5 cells, they're, like I said previously, human lung cells that endogenously express DPP4 and exogenously express CD32A. So overall, Figure 6, it shows the ADE of the MERS-COVID entry. It depends on the range of the MERS monoclonal antibody dosages and on the expressions of the viral and the Fc receptors on cell surfaces. All right, last little bit here. Future studies that they are looking into, they never did any in vivo testing, so that was their main thing that they wanted to do. Based on Figure 4 results, if you remember, it was that image that I said I didn't like and it's really hard to see. It was the EM image. The researchers, they wanted to run a high-resolution cryo-EM in the structure of the MERS-COVID ectodomain trimer complexed with the MERS monoclonal antibody to provide that detailed structural information for the MERS monoclonal antibody 1 triggered conformational changes. So that was step 1 of their future studies. Step 2, they wanted to use in vivo systems to confirm results of these in vitro systems that provide the model framework for ADE. And then for part 3 of their future studies, they wanted to do in vivo studies. And this was basically just to screen for a wide range of antibody dosages and also for a variety of tissues with different ratios of DPP4 and Fc receptor expressions, because a previous study showed that humanized versions of MERS monoclonal antibody efficiently protected human DPP4 transgenic mice from live MERS COVID challenges, which suggested that the receptor-dependent pathway of MERS COVID entry dominates over ADE in vivo. So the whole point of this study basically was to understand the mechanism by which ADE occurs in coronavirus. And at first I was confused because I was like, well, how is this treating coronavirus? But the point was not to treat coronavirus. It was to better understand how the antibody basically allows the virus to infect cells, so they could study that mechanism and make a vaccine that'll prevent ADE and allow us to not get sick again from the same disease but with a different strand. And just a side note, I apologize that this was so long, but there was a lot to talk about, so thanks for listening this whole time.